Hello everyone. Welcome to the new session of Biomolecules past year NEET questions. So in this session we are going to study about some of the questions which came earlier in the NEET examinations. Okay. So I just request you all to prepare your questions based on this session because these are the past year NEET questions. So let us discuss some of the questions in this session. The first question. The amino acid tryptophan is the precursor for the synthesis of. So there are certain uh, substances here, melatonin, serotonin, thyroxine and triiodothyronine, estrogen and progesterone, cortisol and cortisone. So here we are going to study that amino acid tryptophan. This is particular amino acid tryptophan. This is the precursor for the synthesis of. Okay. So let us take the B option. Thyroxine and triiodothyronine. This is the precursor of these two is tyrosine enzyme. If you remember, tyrosine enzyme. Okay. The precursor of progesterone is pregenonine. Pregenonine. This is one of the compound precursor of this. Estrogen precursor is androgen. Okay. So this is also not correct. Cortisol or cortisone. This is the precursor is cholesterol. Cholesterol. Okay. So the melatonin and serotonin, these are the two hormones that precursor is tryptophan. Precursor is tryptophan. So next question, which of the following statement is wrong? Sucrose is a disaccharide. Sucrose is a disaccharide. This is the first statement. Cellulose is a polysaccharide. This is the second statement. Uracil is a pyrimidine. Glycine is a sulfur containing amino acids. So sucrose is a disaccharide. This is the correct statement. Cellulose is a polysaccharide. Yes, this is also correct. We have studied about three different polysaccharide: starch, glycogen, and cellulose. Cellulose. So this is a polysaccharide. Uracil is a pyrimidine. Yes, uracil is found in the case of RNA. Instead of thymine, they are having uracil. Adenine and guanine are the purine, and cytosine is and uracil are the pyrimidines. Glycine is a sulfur containing amino acids. No, this is incorrect. Glycine is do not contain sulfur. Cysteine. Cysteine is a sulfur containing amino acid. So this is incorrect. Glycine is a sulfur containing amino acid. Next question. A typical fat molecule is made up of 3 glycerol molecule and 1 fatty acid molecule. 1 glycerol and 3 fatty acid molecule. 1 glycerol and 1 fatty acid molecule. 3 glycerol and 3 fatty acid molecule. So glycerol plus fatty acid unite and form fats. So glycerol structure is containing hydroxyl group, fatty acid R1CH2 contain acidic group, R2CH2COOH group, R3CH2COOH group. Now this OH and this H comes out, taken out as a water molecule. So 3 water molecule is given out. So here we see that 1 glycerol and 3 fatty acid is required. 1 glycerol and 3 fatty acid is required to form fat. So B1 is the correct answer. 1 glycerol and 3 fatty acid molecule is required. This is a very important question. Next question. Which one of the following statement is correct with reference to enzyme? So if you remember, the enzyme is having two parts. Two parts. The enzyme which is made up of protein. The enzyme which is made up of protein. Okay. That is called as apoenzyme. That part is called as apoenzyme. And sometimes the second part do not contain protein. Sometimes they are having metal ions, they can contain ions and some other prosthetic groups. Okay, that is also called as a prosthetic group. So that is called as coenzyme. That part is called as coenzyme. So apoenzyme and coenzyme unite together to form holoenzyme. That total part is called as holoenzyme. So let us go to this. Holoenzyme is equal to apoenzyme plus coenzyme. Coenzyme is equal to the apoenzyme and holoenzyme. Holoenzyme is equal to coenzyme plus cofactor. Apoenzyme is equal to holoenzyme and coenzyme. So now here we can easily, from this discussion, we can easily make out the first option is the correct. That is, the holoenzyme is equal to apoenzyme plus coenzyme. Yes. Next question. Which of the following are not polymeric? Not polymeric. Proteins, they are the polymers. Polysaccharides, again, proteins are the polymers of amino acids. They are the polymers of amino acid, but they are heteropolymer. They are not homopolymer. All the amino acids are not same, so they are heteropolymer. Polysaccharide, polysaccharide. Sometimes they are, again, they can be homopolymer or they can be heteropolymer. Like cellulose is a homopolymer and chitin is glycogen is a example of heteropolymer. Lipids, nucleic acid. Nucleic acid is again a polymer. You can say a nucleotides. Nucleotides unite together and form a polymer-like structure. But lipids are not a polymer. Lipids are not polymer structure. So correct option is lipid. Next question. The two functional group characteristics of sugar are hydroxyl and methyl group, carbonyl and methyl group, carbonyl and hydroxyl group, carbonyl and phosphate group. So two functional group which are present in the sugar and they are very important. So they are carbonyl and hydroxyl group. Carbonyl group and hydroxyl group. These are the two groups which are present in the sugar. Methyl group is not there. GLUT1, GLUT2, GLUT3 and GLUT4. Which of the following glucose transporter in insulin dependent? So there are different GLUT. These are receptors like present in the plasma membrane that through which the different component get enter inside the cells. So for the insulin, which transporter is having this GLUT4? The GLUT out of these four. So that is GLUT4 is for the insulin. If you remember, this is given in the NCERT chapter. Very important. Concaveline, A is an alkaloid, essential oil, lectin, pigment. So concovalin A is a lectin. It is a very important in the immune system. Okay, lectin. Next question. Consider the following statements. Coenzyme or the metal ion that is tightly bound to enzyme protein is called as a prosthetic group. A complete catalytic active enzyme with its bound prosthetic group is called as apoenzyme. So coenzyme or the metal ion, sometimes the coenzyme, there are different prosthetic groups and sometimes that is present in the form of metal ion. So the coenzyme or the metal ion is tightly bound to the enzyme protein and that is called as prosthetic group. Correct. A complete catalytic active enzyme with its bound prosthetic group is called apoenzyme. No, apoenzyme is just the protein part. Apoenzyme is just the protein part. Okay, metal is the prosthetic group. As a prosthetic group, that is a coenzyme. When joined together, they form holoenzyme. Holoenzyme. So select the correct option. Both A and B are true. A is true, but B is false. Both A and B are false. A is false, but B is true. So correct option is B. A is true, but B is false. Next question. Purines found both in DNA and RNA. Adenine and thymine. Adenine and guanine. Guanine and cytosine. Cytosine and thymine. So what are the purine first of all? Adenine and guanine. These are the two purines that are found in both DNA and RNA. Thymine is not present in RNA. Instead of thymine, uracil is present. So that is pyrimidine. Okay. So adenine and guanine are the two purines that are found in both DNA and RNA. Identify the substances having glycosidic bond and peptide bond respectively. So first of all, glycosidic bond present in polysaccharide and peptide bond is present in protein. So glycerol and trypsin, cellulose and lactine, inulin and insulin, chitin and cholesterol. So cholesterol, type of lipid, and chitin, a polysaccharide. So this is correct, but this is incorrect. So D1 is wrong. Cellulose, glycosidic is there. Lecithin is a lipid. So no peptide bond is here. So this is again incorrect. Glycerol and trypsin. Okay. So this is also incorrect. Inulin is a polysaccharide. Okay. And it is a polysaccharide of fructose present in beta vulgaris. Insulin, it is an hormone which is made up of protein and that contain peptide bond. So this is the correct one. Inulin and insulin. Very important question. Identify the basic amino acid from the following. Glutamic acid. This is a acidic one. Valine. This is a neutral. Tyrosine, aromatic amino acids, neutral. Lysine, this is a basic amino acid. So lysine is a basic amino acid. In the chapter, when we were discussing about the chapter, I have given a bifurcation of all the types of protein, whether it is an acidic, whether it is a basic, whether it is a neutral, whether it is a aromatic, okay, sulfur containing. So if you remember those amino acids classification, so you are able to answer such type of question. So please remember those types of amino acid, okay, then only you are able to solve such a questions. Moving to the next question, which of the following statement is correct? Adenine pair with thymine through one hydrogen bond, adenine pair with thymine through three hydrogen bond, adenine does not pair with thymine, and adenine pair with thymine through two hydrogen bond. So adenine always pair with thymine and guanine always pair with cytosine, or vice versa. Thymine always pair with adenine and cytosine always pair with guanine. This is universal. Now, how much bond is present here? So if you remember, when we were discussing about the nucleic acid structure, then adenine and thymine are having two hydrogen bonds in between them, and guanine and cytosine are having three different hydrogen bonds. So adenine pair with thymine through three hydrogen bond, this is the correct. Adenine pair with only one hydrogen, this is incorrect. Adenine does not pair, this is totally incorrect. And adenine pair with the thymine through two hydrogen bond, adenine pair with thymine with two hydrogen bond, this is correct. This is incorrect. Adenine pair with thymine with three hydrogen bond, this is incorrect. So the option D is correct. Adenine pair with thymine through two hydrogen bonds. Next question. Identify the incorrect pair. Drugs, ricin, alkaloids, codeine, toxin, aberine, lectins, concovalin. So alkaloids, codeine is an alkaloids. Okay, so this is correct. Toxin is aberine. Lectin is a concovalin. Yes, it is a correct. Ricin is a drug. This is incorrect. It is actually a toxin. Okay, so this is incorrect pair. Okay, moving to the next question. Which of the following are not secondary metabolites? Remember, secondary metabolites. So this is about in plants. So rubber and gums, these are secondary metabolites. They are not formed primarily. Okay, morphine and codeine, these are all again secondary metabolites, alkaloids. These and alkaloids are secondary metabolites. Vinblastine and curcumin, these are again secondary metabolites. Metabolites. So rest is amino acid and glucose. These are the primary metabolites. Okay. So glucose is formed as the process of in the process of photosynthesis. Amino acid is formed by the plants only. So these are a primary requirement of the plant. So they are primary metabolites. Okay. So the correct answer is amino acids and glucose. Next question. A dehydration reaction take links to glucose molecule to produce maltose. If the formula of glucose is C6H12O6, then what will be the formula of maltose? So two glucose molecule. So C6H12O6. C6H12O6. Two molecules of glucose is here. Okay, and one water molecule is come out to form a bond in between these two and form a maltose molecule. So you take out H2O here. So total hydrogen, how much? 12 and 12. That is 24. And out of this 24, I am writing 22 because 2 has been removed out in the water. How much oxygen is there? O6, O6. That is 12. And out of 12, 1 is removed out in the water. So I will write 11. C6, C6, that is C12. So C12 H22O11 is the formula of maltose. So the correct one is C12H22O11, the formula of maltose. Next question. Cellulose does not form blue color with iodine. Why? Why? Starch form blue-black color when treated with iodine, right? So you must remember the structure of cellulose and starch. It is a disaccharide. Cellulose does not form blue color because it is a disaccharide, according to this statement. It is a helical molecule. It does not contain complex helices and hence cannot hold the iodine molecule. It breaks down when iodine reacts with it. So if you remember the structure of cellulose, it is a linear chain-like structure and having a cross-linking in between. So there is a no helic-like structure. And it is a very complex structure. It is not a disaccharide. It is not a disaccharide. It is not from helical structure. It breaks down when iodine reacts with it. This is also incorrect. When you put cellulose, put iodine in cellulose, iodine do not react with and it do not break down even. No such thing doesn't happen. So this statement is totally incorrect. Now comes to this. It does not contain complex helices. Yes, cellulose do not contain complex helix like the present in starch. First carbon will form a bond with sixth carbon. First carbon form a hydrogen bond with sixth carbon. Okay, so that forms the helical structure, and in this helical structure iodine get attached and form a blue-black structure, blue-black complex structure. So that occur in the case of starch. That doesn't occur in the case of cellulose. So it does not contain complex helices and hence cannot hold the iodine molecule. This is the correct statement. This is the reason. Okay. Next question. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of. So malonate is a inhibitor. So succinic acid or succinate, it is converted to fumarate in citric acid cycle. If you remember, citric acid cycle. In the presence of enzyme succinic dehydrogenase. And malonate comes and join with succinate in place of succinic dehydrogenase. So this conversion is stopped. Okay. So malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of succinic dehydrogenase enzyme. So in this question, we are going to have a match. Inhibitor of a catalytic activity. Okay, possess peptide bond, cell wall material in fungi, secondary metabolite. So inhibitor of catalytic activity, in the previous question we have just seen malonate. Possesses peptide bond and cell wall material in fungi. So chitin is having a cell wall material in fungi. Secondary metabolite. ricin, and possesses peptide bond collagen protein. So this is the match. So correct option is this. Next question. Match the list 1 and list 2 protein. Unsaturated fatty acid, nucleic acid, polysaccharide. So protein is having peptide bond, unsaturated fatty acid, CC double bond, nucleic acid having phosphodiester bond, and polysaccharide will have a glycosidic bond. Okay, so you can have a correct match as a B1. So this is all about this session. We will meet with one more interesting session. Till then, thank you very much.